Am I audible? Hello?
Yes, sir.
Okay.
Audible.
Thank you. So today we will learn about half an hour about this last topic, that is the especially the animal toxicological test for the study of metabolism, synergism, and antagonism. There are varieties of the laws and other things. We have yesterday or day before yesterday we have heard about the protocols, especially what are all the main definitions of this metabolism, synergism, and antagonism with a basic background. That's what it is required. Today, we'll directly go to the experiments as such and what are all the guidelines as in this required, which are all the OECD guidelines as, and what is the importance of this study of metabolism, synergism, and antagonism. So, coming to the first slide, that is the livestock metabolism. This metabolism study is also called as the livestock metabolism studies. So this discover the regulations required by the pesticide producers to demonstrate that their products present with minimal risk to the health of the humans and animals. That's why it is also called as the drug residual effect or pesticide residual effect. That's what it goes with. So varieties of the types of the studies are there. So this is studies are by the Organization of Economic Cooperation and Development, have two specific test guidelines as for the animal metabolism and residues testing in case of livestock, which are harmonized across most of the countries of the world. Means there are two types of the tests, especially OECD 503 and 505. In the left and right side of the window, you can see this thing. And this is harmonized, means uniformly this is accepted in many parts of the world. So coming to the guidelines, as the title is OECD E503 and 505. So the guidelines as title deletes, this is the metabolism in the livestock and residues in the livestock. So metabolism is totally different from that of the residues. So metabolisms are studied by the 503 guideline, whereas the residues are studied by the 505 guidelines. So the date of adopted is January 18 2007, both are adopted. And the purpose of the first one, that's the OECD 503, is to provide the estimate of total residues in the edible livestock commodities as well as the other parts, and also to identify the major components of the animal residues in the edible tissues. That's what, thus indicating the components to analyze the residues quantification studies. Especially the definition of the residue is totally different, whereas the OECD guidelines as 505 denotes the basis of establishing maximal residue limits, or it is relating to the residue concentration, and for conducting dietary intake assessment of the consumer safety. Whereas the test animals in 503 are typically lactating ruminants. Please note that the lactating ruminants includes goats and the laying hens, especially the poultry, whereas the OECD test guidelines 505 says it is typically the lactating animals, the residues in the milk, especially of the dairy cows, and also the laying hens. So the test molecule is usually radio labeled in 503, and parent compound is also, or its metabolites are radio labeled. They are administered to the animals, and the residual effects are seen either in the meat or either in the milk or also in the egg. Whereas in case of OECD 505, it tests parent compounds or the plant metabolite is also specific, and it is available in the respective links, especially the OECD guidelines as www.oecd.library.org. You can go to that particular website and download this guidelines and go in detail. Whereas I have tried to summarize these guidelines as in very simple steps so that you can understand it very easily. So the livestock exposure to pesticide results from three situations. Number one, the application of a pesticide to raw agriculture commodity or RAC, which is fed to the grazed by the livestock. Second one, direct pesticide application to the livestock. For example, that is to treat or the tick or mite infestations in case of the cattle. So now this synthetic pyrethroids like this flumethrin, deltamethrin, etc., they are applied. And the third one is application of pesticides to the premises of the livestock household. For example, treatment of lice, especially in the poultry shed, and also tick application, especially in case of the cattle directly to the manger, etc., and also to the chickens to their nest boxes and litters, because this it's a menace, especially the especially tick mites, etc. They are a very big menace to the livestock and poultry industry. Hence, it is very important for the estimation of pesticides residues, and this situation arises. So coming to the animal toxicological test for the study of the metabolism. So this is especially the metabolism is noted by OECD guideline 503. So here metabolism in livestock studies, as the title itself is the metabolism in livestock studies. So these are used to determine the qualitative and quantitative metabolism or degradation of the active principles resulting in pesticide uses in the feedstuffs or direct application to the animal premises or the manger or to the place where the animal is always resided with. So metabolism in livestock studies are generally carried out in lactating ruminants and poultry, as we have described this thing in the direct first or two slide. The scientific techniques used to study this xenobiotic metabolism and to conjugate formation, isolation of animal these macromolecules and procedures for generating these monomers they are all advancing, not only because once upon a time this when this guideline was introduced in 2007, the techniques were totally different, and now the techniques are totally different and it is advanced. So it is the responsibility of the applicant to utilize the state-of-the-art techniques and provide citations such techniques whenever it is used. So instead of these HPLC, one may use the LCMS or GC, etc., the major techniques or the more better techniques. So these need to be continuously updated. And this is the OECD guideline for the testing of the chemical 503. It is adopted by these OECD countries on 8 January 2007. So this gist of this thing I am going to tell in the next few of the slides. The purpose of this animal toxicological studies for the metabolism is to provide an estimate of total residues in the edible livestock commodities as well as the excreta also. Here, excreta means a dung or litter in case of the poultry. And to identify the major components of the terminal residues in the edible tissues is also very important. Especially these need to be quantified. That is, the definitions of both risk assessment and enforcement. How to analyze this matter is also an important matter. And to elucidate a metabolic pathway for the pesticide in ruminants and also in case of the poultry, then provide evidences whether or not a residue should be classified as fat soluble, because the fat soluble materials or any other drugs will follow a different type of the metabolism. And the metabolism in livestock studies are needed to elucidate the absorption and disposition of active ingredients wherever a pesticide use or may lead to residues entering the human food chain. Once again, it will enter the human food chain. Whereas in addition, in vitro data are useful to show, I was mentioning you in the earlier classes that the in vitro toxicological data is very important to undergo the hydrolysis. For example, acid, base or enzymatic hydrolysis, then oxidation, reduction, and photolysis or other changes. These need to be studied. So in general, separate metabolism studies should be conducted for ruminants and poultry, because the metabolism process in case of ruminants and poultry are totally different. Of course, the sheep and goat or the cattle, they may have the simple resemblance, whereas it is totally different in case of the poultry. The species of choice are here lactating goats and chicken. Please note that the lactating goats and the laying hens. And non-ruminant, example, many times these swine are also slaughtered, then the metabolism studies may be necessary if rat model metabolism is significantly different than that of the ruminant or poultry metabolism. So this needs to be established. For example, if a drug is discovered, if the metabolism is totally different from that of the rat and also in case of the ruminant, then the study need to be conducted in case of ruminants only. Such differences may include, but are not limited to the following: differences in the extent of the metabolism, number one. Number one, differences in the nature of the observed residues. Number two, because this may totally differ in case of rat and the ruminants. The appearance of the metabolites with substructures which are known potential toxicological concern. All these are the more important. So here, whenever an animal or the poultry or the swine, if it is undergoes a ingestion of the toxicants, then its entire body may contain, or it is distributed or metabolized, and the metabolites may be present in hair, then especially the rumen, then the sweat, and also the saliva. Of course, the muscle tissue or the what you call as the milk or the egg are also important, whereas in case of the pig, it is totally the meat. So there are varieties of the keys for understanding of the conducting metabolism in livestock studies they are complex, and we need to plan and conduct, because the isolation and characterization of active substances and their metabolites can be technically, it is challenging. That's very much challenging. And the regulators require the application of latest thinking and analytical techniques is very much required. Optimization of animal welfare must be considered always, and studies must be designed with timely key decision points to maximize their efficiency and cost effectiveness. So the last line is very important for us, because everybody says that there are the residues, especially in the milk, meat, or the egg. But how to analyze this thing? These are in ppb concentration, and many times these are below the accepted daily intake, or MRL, maximum residual levels, are there. Then there are varieties of the strategies are there, especially for identification and characterization of the residues in all this tissue. So usually the relative amount should always be less than 10%. That's in the first column, whereas the concentration when there is it is less than maybe any of the chemicals is 0.01 milligram per kg of the meat, milk, etc. Milk especially, then no action or no toxicological study should be conducted. Not necessary if it is there, or else if it is in the range of 0.01 to 0.5, characterize only attempt to confirm identity if straightforward. Example, a reference compound is available or identification is known from the previous study. Whereas if it is more than 0.05, then characterization, identification need to be decided on a case by case basis. That's also it is possible, and need to be designed in such a way that we should be able to find the highest level of the concentration. Then if it is less than 0.01, then characterize only attempt to confirm identity if straightforward. And if it is this concentration is ranging from 0.01 to 0.5, that means more higher concentration, significant or marked attempts to identify should be made, especially if needed to establish the pathway. Ultimately, characterization must be accepted. And if it is more than 0.5, identifying use all possible means should be conducted. And if it is more than 0.05, unextracted or it is a radio level, then extractable radio level, further extraction procedure should be exhausted. So varieties of these analytical techniques are there to do the residual analysis of these drugs or the chemicals. So there are two different chromatographic techniques are there. They must be understood before this thing. The techniques such as HPLC, high performance liquid chromatography, are usually preferred, or reverse normal phase TLC. That's TLC is also have got this greater sensitivity. But Unambiguous identification cannot be obtained through the methods that provide structural information such as the LCMS or liquid chromatography, mass spectrometry, that's the LCMS, and liquid chromatography tandem mass spectrometry, that's LCMS or MS. So both of these equipments are there in Veterinary College, Abbala, Bengaluru, and we have analyzed varieties of this tissue residues using this LCMS or MS. So the sensitivity of this equipment, of course, the LCMS MS is highly sensitive compared to LCMS MS, whereas TLC and HPLC are also probably they are sensitive. And the nuclear magnetic resonance NMR, then gas chromatography technique GCMS MS, then this LCMS MS are the commonly used metabolomics platform with varying detection limit. Of course, the HPLC is still the gold standard. If you can see that sensitivity pattern is more in the LCMS MS, and the NMR is very highly sensitive, followed by GCMS, which is with quadrupole, and GCMS or TOF. So the sensitivity is more and more with the cost of the equipment is very, very high, especially NMR and LCMS MS, etc. So how these studies are conducted? So the dosage of a chemical, suppose if you want to know the cypermethrin concentration, especially in a tissue, the milk, etc., then minimum dosage used in livestock oral metabolism studies should approximate the level of exposure expected from feeding up of treated crops with the highest observed residues. So here we have to do the analyze in the treated crops. So in case of dermal application of the minimum dose should be maximum concerned from the label of the particularly a compound. So here the exaggerated dosage are usually needed to obtain sufficient residues in the tissues many times whenever it is anticipated, and also for the identification. Then regardless of the oral studies, livestock should be dosed at least a level of 10 milligram per kg in the diet, and animals should be dosed only orally. Then ruminants and swine should be dosed daily for at least 5 days, thinking that it is consumed for 5 days, and poultry for at least 7 days. So please remember these parameters as per the OECD guideline 503. Then discussion of the test method, especially dosing radio level pesticide is also sometimes it is used. So radio labeled active ingredients are required to allow quantification of the total extractable and unextractable, especially from the residues. So the active ingredients should be labeled so that the degradation pathway can be traced as far as possible, because this is the metabolites may sometime they may be also active. So the radio label should be positioned in the molecule so that all significant moieties or degradation products can be tracked. Suppose you are going to administer the norfloxacin, simply estimation of not norfloxacin, enrofloxacin is not sufficient. Enrofloxacin is not sufficient. You need to estimate the metabolite, this thing, that's the ciprofloxacin. It's converted to ciprofloxacin. Both should be labeled and both should be administered. If multiple ring structure or significant side chains are present, separate studies reflecting labeling of each ring or side chain will normally be required if it is anticipated that cleavage between these moieties may also occur. That's why both of these are very much required. And animal toxicological tests, especially dosing for radiolabel pesticide, require scientifically based rationale. It is very much required because one need to know the metabolic pathway very correctly, or else we cannot say how it is going to cleave, or it need to be anticipated. So the cleavage of the molecule is evident that the applicant may require to conduct an additional study with radio level that tracks the portion of the molecule that is cleaved. Suppose there is a metabolite is also active, then that need to be labeled. Then because one should know the chemical structure of a substance very properly, then the preferred radioisotopes are the carbon 14, then 32 phosphorus and sulfur 35. may be more appropriate, and carbon chain, whenever most of they think they contain carbons, and the tritium 3H is a label is strongly discouraged due to the possibility of hydrogen exchange with endogenous material. That's why the tritium binds very properly with most of the biological material, but it is discouraged because the possibility of hydrogen exchange with endogenous material. That's why it is discouraged. So the dosing of radiolabel pesticide is specific activity of the radiolabel ingredient should be adequate to meet the data requirement of the livestock metabolism. That is the quantification studies, maybe in case of this quantification of 0.01 milligram, especially in the tissues, milk, or the meat. So the use of stable isotopes such as this 13 carbon, 15 nitrogen, 2D unexchangeable together with the radiolabel isotope to aid the identification of the metabolites by various spectrophotic method. For example, mass spectrometry or the nuclear magnetic resonance (NMR) is encouraged. So when the dose is administered orally, livestock should be dosed via a balling gun or capsule or gavage to ensure that complete administration of the active ingredient is there. The dosage administered should be guaranteed before we are going to analyze the residues. So there is a sacrifice is required when to do this thing. One of the main objectives of livestock metabolism studies is to determine the identity of the residues in the edible tissues in case of cattle, sheep and goat, and milk and eggs, especially after the sacrifice. So the applicant will be asked to provide the scientific basis which justifies the time of sacrifice chosen for the species used in the studies. So this time should be preferentially 6 to 12 hours after the last dose. So this is one thing. And suppose we are administering 5 doses, and after 5th dose, 6 to 12 hours, the gap should be there. And no circumstance, the time of sacrifice should be more than 24 hours. Then the excreta, milk and eggs should be collected twice daily. This is applicable. Tissues to be collected should include at least muscle, loin, flank muscles in case of ruminants, and leg and breast muscles in case of the poultry. Of course, the liver, whole organ of the goat, poultry, and representative parts of the different levels, lobes of the liver of the cattle or swine are very much required. And also in case of kidney, then fat, etc., in case of the ruminants. So the sample should be quantified in all tissues, excreta, milk, eggs. For example, the milk fat fraction should be separated because many times the fat is totally maybe adhered with that of the chemical. So the aqueous portion by physical means and by this TRR, each fraction modified. And characterization of this residue in urine, feces subsequently needs to be level, and that should be lower than that of the other tissues. Then radioactivity in the different muscles and fat types should be quantified separately with the specific tests. Whereas the analytical phase, especially the livestock metabolism study, the animal parts should be analyzed. They are sampled, chopped, or homolyzed particularly, and full accountability of all radioactivity should be ensured. And especially this carbon, etc., they are not so dangerous to the human body. And identification refers to the exact structural determination of the components of the total radioactive residues. So one need to do these things. So how to establish this thing is typically identification is accomplished either or co-chromatography by the metabolite. So we need to establish the identification of the metabolite along with that of the original drug. And this need using two dissimilar systems or by techniques capable of the positive structural identification such as the mass spectrometry or nuclear magnetic resonance, very highly sensitive equipment. So in case of the co-chromatography, chromatographic techniques utilizing the same stationary phase with two different solvent systems are not considered to be an adequate two-method verification of metabolite identically, since the methods are not independent. And identification of the chromatography should be obtained using two dissimilar analytically independent systems such as reverse and normal phase thin layer chromatography, RTLC or HPLC system. Two systems should be there for cross-examining this system. So the characterization refers to the elucidation of the general nature, characteristics of the radioactive residues, short metabolized characterization, and the terms used to characterize include the organosoluble or water soluble, neutral acidic, basic, polar, non-polar, non-extractable, etc. Because a material may be having all these nature, and the characterization may also involve descriptions of the chemical moieties known to be present in the molecule based on the conversion to a common structure or to due to the real reactivity of a particular reagent. So instead of, you can also do the different fractions, maybe the organic solvent solution, water solution, etc., or else you can convert it into a single structural chemical, so that the ease of the identification. So the degree of characterization refers how close assignments come in structural identification. So here there is a metabolism studies, especially it is I see exaggerated feeding levels are employed, and low radioactivity results in the tissue characterization, identification, etc. For example, if anticipated dietary burden of livestock is about, in the first chart we have seen that 0.01 milligram per kg or 10 milligram per kg radio level compound is fed. That's the 1000x. The total radioactivity in tissue, milk or eggs is less than 0.1 milligram per kg, and the minimal characterization and identification of the residues should be adequate. And such situations often rise to early season herbicides having very low application rates. So these are all certain things which need to be considered while we are doing the radioactivity assay of 0.01 milligram or less in the particularly that table, and the TLC or the HPLC systems are need to be standardized thing for this thing. So this differs from one concentration to another concentration of the tissue residue level. So here there is a flow chart, how characterization and identification of non-extractable and bound residues. So here you can see that unextracted radiolabel material, if it is less than 0.5 milligram or 10% of the residual concentration or greater, then no additional characterization is required. That's as per the guidelines. Levels down to 0.505 parts per billion or 10% of the residual level, which is greater, then you have to go for varieties of this characterization. So here it need to be mixed with 6N acid, maybe 10N base reflux, then sonication need to be done, or and then final hydrolysis product or the pesticide if it's a pesticide, or else the material need to be sonicated, then enzyme extraction, then labeled sugars, amino acids, etc., then need to be considered, or else they need to be bonded with surfactants for the ultrasonication, then physically encapsulated for the metabolites, or else it need to be diluted with acid or base with the ambient temperature. Then the metabolites and conjugates are formed, and these need to be estimated. So initially, so all these things ultimately there is a effect called as the matrix effect. Matrix means the milk is also a matrix, meat is also a matrix, egg albumin is also a matrix. So these need to be taken care that matrix effect should be as less as possible. That's why we say that matrix extract is throughout the study should be used, and the matrix should not be extracted later in the study, because the stability of the extract should be shown in till the end of that particularly. If any changes are observed, then additional analysis should be conducted. Then These are certain clarifications are also made in the guidelines, especially in the chromatographic techniques, HPLC or TLC. The polarity of the solvent system should be governed by the polarity of the compounds being analyzed. That's the solvent polarity, and clear images of the radio analytical imaging techniques should be maintained forever and ever. Then radio validation of the extraction process should be submitted as part of the report to the regulatory authorities. The metabolism report may be sent also. So coming to that's about the 503 guidelines. Whereas in case of 505, this guideline is somehow, it is especially this guideline is referred to the residues in the livestock, OECD 505. It's also accepted in the year 2007. Then residues in the livestock studies provide data on the qualitative or quantitative transfer or the pesticide residue in the meat, fat. So meat, fat, milk, eggs, and edible meat by the byproducts. So the main objective of these 505 studies are to establish maximum residue limits, that's MRL, in food commodities of animal origin, and to assess the dietary intake of residues in order to assess the impact on consumer safety. So there is no much difference between the 505 or 503, whereas in the case of 503, the metabolism studies are also conducted, whereas in case of 505, only residual analysis is being done. So residues in the livestock studies, sometimes called as the residue transfer studies or the feeding studies, are generally required when quantifiable pesticide residues occur in the feed crops at levels that could theoretically lead to residues more than 0.01 milligram per kg. Then in such case, the animal tissues need to be examined in the way of 505. So situations when these studies may not be necessary is these conventional residues in the livestock studies may not be necessary for the following situations. When based on crop field trials, if the residue levels are below the limit of quantification, LOQ means this cannot be quantified at all, then we need not go for estimation of this kind of material in the animal tissues. Then because whenever there is a intake is very less, there will not be any residual effect, and if this shows no significant pesticides in the bioaccumulation in the animal tissue. So when metabolism study with dosing levels 10 times the anticipated dietary burden shows that residues from the concern are below the LOQ or the typically 0.01 milligram per kg all edible tissues. So in such circumstances, no quantifiable residues should be anticipated in livestock commodities, because unnecessarily we need not go for any of such study. However, without residues in the livestock study, the maximum residue limit need to be estimated based on the LOQ validated for the analytical method for determining the residues in the meat, milk, and also in case of these eggs. So when treatment is applied to animal premises, it may be possible to present scientific justification for not needing specific feeding studies based on data from direct animal treatments, and hence general handling of the animal husbandry practices should be followed. So if it is like that, then the study should be conducted according to 503, assess the metabolism in livestock to determine the qualitative and quantitative metabolism or degradation of the active ingredient of the pesticides. So then expected. Whereas studies are performed in accordance with OECD 505, provide the data on the quantitative, remember it's a quantitative transfer of pesticide residues into the milk, meat, and eggs and edible meat by the products. The main objectives of these studies are establish MRLs in food commodities of animal origin and to assess the dietary intake of the residues in order to assess the impact of the safety. That's how the difference between the 503 and 505 need to be studied. So there is a global, this is the chart, according to a study, the global pesticide residue market by the region during the year that's estimated 2009 to 2007 is, so it is anticipated, it's anticipated that it will go very high, especially in most of the advanced developed countries like North America and Middle East, Africa, etc. You can see that now we are in the 2022, it's anticipated. So till they have estimated a forecast, the use of this material, and that's how it goes. And coming to the summary of this chapter, metabolism in livestock and residues in livestock. Studies are required. See, metabolism is totally different. Absorption, distribution, metabolism, what you have said. So metabolism, studies, and residues, because this metabolism and residue. So after metabolism only, the residues will be there in the animal tissues. These are required by the regulatory authorities in the worldwide in order to understand how pesticides consumed by livestock may accumulate in the edible part of the animal. That's how we need to establish this thing. Accordingly, the OECD guidelines as 503 and 505 are designed, and you have to understand them very properly. And when you become the study director or the advisor in the especially in a regulatory authority, you can guide the respective government properly. That's about this thing is there. And thank you for staying with me. Thank you.